Hello, everyone. So today I'm going to be reviewing the anatomy and pathway of brainstem auditory evoked potentials, and my name is Genesis Huertas. So here's an overview of how sound travels through the ear. It enters here through your external ear, it travels through this canal, hits the tympanic membrane, setting the ossicles in motion in the middle ear, which eventually causes this fluid to disperse around the cochlea. So, first, I'm going to talk a little bit about the outer ear. The outer ear is comprised of the pinna, which is your external ear, the concha. As you move down, you'll see the external auditory canal, and then the tympanic membrane towards the end. There's two functions of the outer ear. It is able to amplify sound pressures of sound frequencies around 3 kilohertz, which is the frequency of speech. So it is actually able to amplify these pressures around up to 100 times, which I thought was interesting. Another function of the outer ear is that it is able to filter sound frequencies and provide cues about the elevation of the sound source. Now we have the middle ear. The middle ear is a air-filled space just behind the tympanic membrane, and this is what separates the outer ear from the middle ear. As you can see, there are 3 tiny bones located in the middle ear, the malleus, incus, and stapes. Together, these are called the ossicles. Down here, you'll see the eustachian tube. The eustachian tube is connected to the back of the throat and the nose, and it is used. Its function is that it equalizes the pressure of the middle ear, allowing the tympanic membrane to move freely. Next, we have the oval window. The oval window is where the stapes is attached to, and it's what causes the fluid of the cochlea to move whenever these movements are transferred onto the oval window. And lastly, we have the round window, and that's where these movements are able to dissipate. So next we have the inner ear. The inner ear is comprised of the vestibular organs here and then the cochlea. The vestibular organs are comprised of the three semicircular canals and a vestibule, and is used mainly for equilibrium and balance of the body. And then here we have the cochlea, which is used for hearing. If you take a cross section of the cochlea in this image in the top left, you will see 3 compartments: the scala vestibuli, the scala media, and the scala tympani. These compartments are filled with fluid, and if you look at the picture on the right, you will see the different types of fluid. So up top here, we have the scala vestibuli, and that's going to be filled with paralymph. And then in the middle, we have the scala media, and that is going to be filled with a potassium-rich endolymph that is important for the depolarization of the hair cells. And then lastly, the scala tympani here in the bottom is also filled with paralymph. So if you take a closer look at the scala media, you will see something called the organ of Corti. And here's a blown up image of the organ of Corti. So the structures in this organ of Corti are the basilar membrane at the bottom, the tectorial membrane at the top, and then we have inner and outer hair cells that have small stereocilia at the top that are important for depolarization. And then the cochlear nerve here exits the inner hair cell and is part of the cranial nerve 8. So what happens is fluid moves around the cochlea, and as the fluid moves around the cochlea, it causes deflections in this basilar membrane right here, which also causes the tectorial membrane to also move. This movement of these membranes causes the small stereocilia on the hair cells to bend, causing their ion channels to open. Like I said before, the endolymph that bathes these hair cells is rich in potassium. So when these gates open, potassium is influxed into the cell and it depolarizes the cell, causing neural impulses to be sent through the cochlear nerve. One thing I found interesting about the cochlea is that it's actually tonotopically organized. So you will see that high frequencies are detected more towards the base of the cochlea, while lower frequencies are detected more towards the apex of the cochlea. So next we have the vascular supply of the inner ear. So perfusion mainly comes from the vertebro-basilar artery, the anterior inferior cerebellar artery, the posterior inferior cerebellar artery, the superior cerebellar artery, and the posterior cerebellar artery. Here you can see some of the branching happening in this picture. You'll see this big artery is the basal artery. It gives off the anterior inferior cerebellar artery, or ICA, which then gives off the labyrinthine artery. And this artery branches into the cochlear arteries and vestibular arteries. One important thing to note is that any ischemia or changes in this blood supply will result in loss of auditory evoked potentials. Lastly, I'm going to talk about the pathway of the auditory system and then the different sites where the waves are generated that we see in BAEP monitoring. So these axons of the cochlear nerve, which is part of cranial nerve 8, leave the cochlea and enter the cochlear nucleus. The distal portion of these axons, where the hair cells are located, is what makes wave 1. When these axons synapse in the cochlear nucleus, this creates wave 2. From the cochlear nucleus, axons from one ear are sent to both superior olives. So these superior olives receive information from both ears. This creates wave 3. The superior olives are important for the localization of sound. And so what it does is it gets these information from both ears, the sound information, and it uses the temporal and intensity differences of the sound coming in from both ears to determine where that sound is coming from. From the superior olives, axons travel up through the lateral lemniscus, and this creates wave 4. They eventually synapse in the inferior colliculus, which creates wave 5. The inferior colliculus is, it creates a sort of topographical map of the auditory space. So it has neurons that are, that have a preference for certain auditory sounds coming from certain spaces in the auditory space. So it creates a map based on those preferential neurons. From here, from the inferior colliculus, axons travel up to the medial geniculate body of the thalamus. This is what creates wave 6. From the thalamus, axons then travel to the auditory cortex of the temporal lobe, and this is what creates wave 7. And here are my references. Thank you.